I think this generation gets excited about things having to do with biology. This is kind of the century of biological engineering. And one of the ones that just keeps coming up again and again, even for a decade when we were making no progress on it whatsoever, is the woolly mammoth. We give all the caveats, how it's just a cold-resistant elephant and it's many years away, and there's technology to be developed and so forth. It doesn't matter. There just seems to be an insatiable appetite, especially among young people.
Ignition sequence start. 6, 5, 4, 3, 2, 1, 0.
Thanks so much for joining us, Dr. Church. We are just so excited to have you here. This is a subject I'm super interested in as, at one point in time, I wanted to be a geneticist. I went to, I think it was like a pre-college program at the University of Medicine and Dentistry in Newark, New Jersey, where I grew up, and they had some students come in, and I thought it was fascinating. And then when I got to high school, I caught the engineering bug and went down that route. So just so happy to have you here.
I'm happy to be here. It's great talking to you.
Can you talk a little bit about, like your journey and what really inspired you to pursue genetics, and how did your early work shape kind of where you are in your career right now?
Yeah. So you mentioned that you got the engineering bug. I think I had the engineering bug too, but for biology, not for physics and chemistry. And I maybe I got a little bit of it from being, growing up in Florida, not too far from Cape Canaveral. But I think I really got fascinated with, what you could do in agriculture and in medicine. My third father was a physician, and he'd do his house calls with his black bag full of technology, like needles and syringes. He would sterilize in a little baby autoclave, and all kinds of drugs with brilliant colors, like cobalamin. Anyway, so I think I was an engineer, but didn't know it because everybody called engineers scientists back then. It was rocket scientists
Yes.
was the term for clearly people who are engineering rockets. Yeah, so then I was getting a pretty lousy education in Florida, both from science and engineering standpoint, almost every standpoint. But then fortunately, I got sent away from my family when I was just turning 14 to Massachusetts, and it was really like opening up a whole, cornucopia of ideas and opportunities. And I just, I responded very enthusiastically. Did a lot of experiments on my own, including organic chemistry syntheses, which I can't believe they let me do that on unsupervised at nights and weekends during the 1960s and early 70s, which was a time where young chemists were not always doing reputable things. Um, and then I, continued doing research, looking for intersections of all the sciences, because I really was kind of ADHD and just liked them all. And the first thing I found that really captured all that was X-ray crystallography, and it involved technology development, computer science, math, the Fourier transforms, all the way up to medicine. And that inspired me to go to Wally Gilbert's lab for graduate school, where he was just on the verge of, getting the very basics of DNA sequencing going, for which he got the chemistry Nobel a few years later. And that was again one of these epiphanies, opening up of new doors for me, being in that environment. And then I went to UCSF, where I learned about stem cells, brought that back to Harvard, became a professor, and helped start the Genome Project around 1984, and then did a whole series of, keeping with the ADHD, and went from genomics to proteomics to transcriptomics to functional genomics to systems biology, synthetic biology, immunology, neurobiology. and virology, and of course, mastering almost none of these. But it's still publishing a few papers and a few patents.
You said ADHD, but I think, things are a little bit different now, as to before, because even, for myself, I know, it was like, stay focused in one particular area, you don't want to be the master of none and the jack of all trades. But I think it's so fascinating nowadays, even with my own children and just kind of what you hear, you want to encourage students to really get involved in multiple areas so that they have more options.
I think there's a much larger niche now for multidisciplinary experts, people who can bridge fields. We need a lot more bridging because the disciplines are getting deeper. They're getting so deep they can't see out into the other disciplines. So you need people that will bridge those. That's good. Um, I think for my own work in technology development is particularly useful. And in fact, my team is composed almost entirely of multidisciplinary individuals. We have relatively few true disciplinarians that are entirely focused because it helps to bind the team together to have all these people that can speak multiple languages, metaphorically speaking. So um, it's really a pleasure and it helps you appreciate, the world is going, especially the technological world is going exponentially now. And to really appreciate it, it really helps to have training in a number of fields. And, uh, and I never really jettison any of these fields. I just keep adding them. And it really, it at a minute, we contribute to many of them, but it's also just nice to appreciate them the way you would go to a concert. You don't have to be in the orchestra to appreciate.
Right. Since you said it's more like, multi, disciplinary, how do you think, just from what you've seen from the time you started till now, how technology has really played a huge impact in, just, this particular space and not only focusing just on, genetics and, just the medicine and science of it, but now all of the technology that's really brought in as well.
Yeah, I mean, technology has always had an impact. There was a big burst of technology around the year 1900 when Edison and Tesla and others were doing things with electricity and photography and so on. And then, um, but what we have now is a really recognizable exponential technologies where you can you can monitor the progress in factors of 2 per year, which adds up to factors of 20 million, for example, in reading and writing DNA, not just in cost, but in quality. Um, so something that was, almost incomprehensible, like, when I started, you could barely sequence a few base pairs, and now we think nothing of, ordering up several genomes of 6, billion base pairs each, and, so that exponential has had a big impact, and it almost always spreads out quickly. Just so the kind of technologies I'm interested in are kind of basic technologies. Technologies can be used for many different fields, not even just biology. Some of them are used for, material science, with non-biological,
Mhm.
not conventionally biological materials, um, forensics, you know, history and archaeology. It's really the sweep is vast. And then AI is something that also applies multiple fields, including biology. And so we're kind of at the interface between these two things, AI and biotechnology.
Yeah, so thinking about all of those things and, how technology touches it, what really excites you most about the field of synthetic biology?
Well, synthetic biology, you know, broadly speaking, is like synthetic chemistry is broadly speaking about anything that you're making with chemistry, which is either you're imitating something natural or you're inspired by something or you're doing something completely new. Same thing with synthetic biology. It ranges from making microorganisms that are resistant to all viruses and then trying to extend that to other organisms to make everything that you care about, resistant. To, that's at one end. The other end, you're making, humanized pigs so that they can donate organs. Uh, but it's the same basic molecular biology tools that allow you to do this, you know, materials, virology, antivirus and organ immune um, tolerance.
You talked a little bit about, multidisciplinary and most of the students in your lab really, coming from different areas. Much of your work has really focused on democratizing, science, including creating, more open resources for genetic research. How do you really, envision making advanced, more scientific tools that are more accessible to young scientists and inventors all around the world?
Well, step one in that has been something I've been obsessed with since I was a teenager, is bringing down the price of things. And I didn't realize just how radically that was going to happen. 20 million fold is quite a bit to do in just, a decade or so. Um, but it requires more than that to make it accessible. I mean, you can bring down the cost of, vaccines down to less than $20, less than even $3. But you have to get everybody to understand how powerful it is, how good it is for them. Um, but eventually you get to the point where for certain human-specific diseases, you don't need the vaccines anymore because you've made the pathogen extinct. And for example, smallpox for sure, polio almost there, guinea worm. The same thing goes for, all the other molecular technologies like gene therapy and genetic counseling and so forth. These things require that you enable the less fortunate. Bringing the price down is enough. You have to really have a conversation with them where they get excited about it.
Yeah, I think the other thing too, and just in terms of what I've seen, even myself as an engineer, and I talk a lot about the cloud, and you mentioned AI, just having them to understand how that intersects with other areas of what we call STEM or STEAM now, right? So when I talk to students about that, who may be interested in medicine, having them understand that, some of the technology have really helped, scientists, and researchers be able to discover, genetic mutations at such a faster rate, and this is why, it's really interesting for them to have just some kind of base knowledge and understanding of, like AI, what the cloud is, and how that technology impacts. Are these conversations that, maybe you've had with some of your colleagues or some of these young scientists?
Well, I think the young scientists, by the time they become young scientists, are already often ahead of their elders. So that I think the real challenge is while they're still deciding whether they are going to even take an advanced science course or any optional science or math course, STEAM. Um, so I think that requires that they have role models and exciting projects that they can see from very far away at kind of so I think an example of a project um, that got my generation excited was going to the moon and beyond. um, I think this generation gets excited about things having to do with biology. This is kind of the century of biological engineering. And one of the one that just keeps coming up again and again, even for a decade when we were making no progress on it whatsoever, is the woolly mammoth. We give all the caveats, how it's just a cold resistant elephant and it's many years away, and there's technology to be developed and so forth. It doesn't matter. There just seems to be an insatiable appetite, especially among young people. And so I take that seriously. I say, let's not engage them on all the other topics they're not interested in until it kind of organically transitions into that conversation. And also let's listen to what they're interested in. They might actually be seeing something about the future that's important. Certainly conservation, endangered species, restoring the Arctic, these things that they're seeing are intuitively appealing. may also be feasible and so forth. So I think it's good to both listen and educate. They educate us as much as vice versa.
A lot of your work as well on, like de-extinction has really captivated the public's imagination. What are the scientific and, ethical challenges of bringing back extinct species? And how might, this research, really benefit biodiversity and the ecosystems?
Many of these species, humans contributed to their downfall. We had a voracious appetite, and we would often kill, animals just to have a snack, and then the rest, and so I think most of the major herbivores in the Arctic, and even a little bit south of that on all the continents, disappeared around 10,000 years ago, thanks in part to us. You can blame it on climate change way back then, but I think the big difference was the appearance of humans. Um, so the ethics and the scientific challenges you asked about are, we don't necessarily want, even if we could, and I think we will very soon be able to make a perfect genome from scratch. I mean, we can already make genomes from scratch for simple organisms, but we should be able to make it for any organism soon. But even if we could, I'm not sure from an ethical standpoint, we would want to. We would want to make something that's perfectly adapted to modern world. Is providing something that humans care about, so that humans will be tempted to nurture the animals rather than kill them off again. So from an ethical standpoint, we want to tip the balance towards, for example, giving elephants, which are endangered species because they're in close proximity to humans, give them some land in the Arctic where they're really at very, very low human density, and maybe a more conciliatory attitude as well. Maybe reduce their tusks at first so they won't be that won't be a target for
Mhm.
them. Um,
Yeah.
be nice if you could get rid of the rhino horn and the shark fin too, because that puts them at risk. Because people will harvest them just for those little things,
Yeah,
the tusks, the,
exactly.
the fins, and the horns. And but in any case, make them less accessible. Give them a purpose, which is restoring the vibrant ecosystems that sequester carbon much better than they do today. Um, and I think that and then the final technical component of that is we need better reproductive technologies for endangered species, in particular, endangered mammals, in particular.
Can you talk a little about the research that is being done to really bring back the extinct species?
Right. Well, some species can be brought back almost trivially because they were frozen or stored in a suitable cold environment. So there are some plant seeds that survive for millennia, and they can be restored that way. There are a few animals that were frozen at the dawn of proper refrigeration, but these are exceptions, and they're usually pretty recent species. They're not ancient ones. To go further back, you can go back probably 2 million years via DNA. Now, DNA is a convoluted step because you have to read it into a computer, and then you have to use the computer to write it again, and then you have to get it organized in the right way. You might have to add a few changes that make them adapted for the modern world, like, say, avoiding, you know, highly toxic viruses that are specific for Asian elephants. So you've got this cluster of things that you need to do.
You mentioned also, helping to really start the Genome Project. Can you, just talk a little bit more about that for, people listening, and just, really describe what the Human Genome Project is and where it has led?
Well, going backwards from where it has led, part of the reason that we have now sequences of almost every major category of plants, animals, and microbes is because we brought the cost down by 20 million fold. And that applies not just to the genomes, but to their, you know, the RNAs, the way they develop, all the different cell types in their body and so forth. So backing up from that 20 million fold reduction in price, we had initially a project that was just humans. At least that's the way it was initially framed, and just $3 billion. And I was not thrilled with either one, even though I was there in a small group where we could propose this. I felt it should be multiple species and technology first. Before we spend a lot of money, let's bring the price down as to a reasonable price, which I thought was probably less than $1,000 for human, and probably much less than that for bacteria, since they're about 1,000 times smaller genomes. So, and that did happen, but it happened after we spent the $3 billion rather than before, which was not, it was I was a little too young to have much influence, other than said my opinion. But I just used the time to develop the technology, and so it was ready as soon as the Genome Project was over, and then that technology, it started out by what was called gel electrophoresis, and it transitioned to essentially microscopy. We use microscopy to pack in the information into little fluorescent dots of light, and that was millions of times more cost effective. It was around 1984 when we started thinking about sequencing genomes. When I first learned about genomes or even genes back in
Mhm.
the 1970s, early 70s, the idea that you could sequence even one was challenging, and sequencing all of them was, laughable. Not by
That
me,
is
but a
so,
lot of other people.
but a lot of other
Uh,
people.
yeah.
You mentioned as well, just bringing back ancient plants that sequestered CO2 better. Can you talk a little bit more about this and your role in this?
Well, it's not so much ancient plants. We do have, along with the 2 million-year-old uh, mammals, we also have 2 million-old bacteria and plants, but they're not that different from the modern ones. So we have actually plants today that are capable of amazing carbon sequestration, more amazing than typical farm plants or trees, typically grasses. And the thing that you want to bring back is not the genome of those ancient plants, but the ecosystem. So, what happened is, as soon as our ancestors, killed off all the mega herbivores, then that which were keeping the trees in check, the trees took over, and they kind of squeezed out the grasses, and the trees result in much thicker, fluffy, insulating layers of snow, which resulted in warmer um, permafrost, which can result in release of carbon in the form of methane and CO2, which are very serious global warming gases.
Yeah.
So, so it was just not appealing, both from a ecosystem conservation standpoint, as well as modern human needs for uh, overall climate.
And, just thinking about, the future of genome editing, I know that, the term like CRISPR has become, more of a household term in the world of genetics. What are some of the most really impactful applications of genome editing?
Yeah, so my lab was working on genome editing before CRISPR, and then we helped get CRISPR to be a technology, and then various technologies that are un-CRISPR related since then as well. But the key component of all was editing, and in particular kind, which is um, multiplex editing, meaning changing more than one gene at a time. So most simple diseases, all you have to do is change one gene at a time. But those same simple diseases, which can be quite life-threatening at a very young age, can be handled much more cost effectively using genetic counseling, which is another benefit of the genome sequencing revolution, where you have a choice. whether you do it by editing, which costs $4 million per person, or via counseling, which costs, $400. So to me, that's a pretty clear choice, but that hasn't been clear to the whole world yet. Um, so the other things you can do with editing, aside from simple one gene at a time, is age-related diseases. So we have a few things going into clinical trials there. And there, it's not editing in the usual CRISPR sense. It's just bringing in genes that have where the expression has gone down with age. You just boost them back up by bringing in. And one of the advantages of doing that kind of gene therapy is it's once and done. You give, or at least it's done for as long as the first dose. The first dose you got from birth until age 70, then you get another dose, and you're good for a few decades more. So aging is one thing. Organ transplant, there we had to do multiplex. So we need multiplex editing for aging and multiplex editing for transplants. 69 genes we had to edit for transplants. We had to do 3 to 6. For aging, we actually don't know how many it will eventually be, probably much more than that. Another thing is the de-extinction. There's probably hundreds of genes we need to change, even just to get the ecological, functional benefits, and millions if we wanted to make an exact copy, which I don't think we do, for ethical reasons as well as technical ones.
Yeah, I was gonna ask about, what do you see, some of the ethical considerations really playing in this development?
Some of the ones that are raised are, well, are we bringing back something that died just to die again? And if we bring back an exact copy, yes, we may be setting ourselves up for that. But if we bring back, a copy that's better adapted, so for example, for elephants, we are in the process of making them resistant to, an extinction level virus called EEHV. So that's a whole new game. That's not bringing back an exact copy. We might make them resistant to poachers by making their tusks a little bit shorter. We might fine tune their summer winter adaptations. It's still cold there. I mean, people say that it's not cold in the Arctic should spend a winter there. Even a lot of my
Oh,
friends
I'm sure.
who live in Siberia during the summer do not live there in the winter. So it's like -40, -30.
Oh
Um,
my goodness.
but nevertheless, fine tuning the temperature in the summer and winter, and um, you know, uh, dealing with mosquitoes and mosquito-borne infection. That's another thing that can be done. So there's just a lot of things that we can do to make the life of endangered species so much better. Um, and I think that's one of the major goals of this sort of project.
Mm. So, I mean, I know you speak, countless places at events and conferences, but I know that you recently spoke at Oxford at an event regarding human enhancement. Enhancement. Can you talk a little bit about this and what are some of your thoughts on the future of human enhancement?
Well, this is specifically having to do with sports, and I think it's well known that many sports use enhancing drugs, without permission. And so you have kind of the disadvantages, sort of the ethical disadvantages, that some people who are following the rules are at a disadvantage. That's not good. Some people who are not following the rules are putting themselves at health risk because they're not being properly monitored because they're trying to keep it a secret. So and also just to note that sports probably started out as a technology development, augmentation event. So, you started out and you would run without shoes. And so shoes were something that put you at a huge advantage over people who didn't have shoes. And, same thing, spears and discus and all these things were ways of, javelins, were ways of, practicing for the hunt, and developing technologies. So there was no real rules. You just wanted to be able to be better hunters. So fast forward to today, and it would be nice to have a controlled environment where there's more openness. People aren't fearful that if they, if a little bit of of an illegal drug gets detected in their urine or blood, they're out. That's it. They're done. It would be much better to have a system where you're monitoring it. You're just doing like clinical trials, um, in a way where you can exchange information and learn from. Um, both the good and the bad. So, that's what attracted me to this concept, and I think it's really gaining momentum very quickly.
What was the event called, that you spoke at Oxford?
It was sponsored by the Enhanced Games. It was on that topic of enhancement and alternatives to having all these enhancements hidden, where it's risky and you don't accumulate knowledge as quickly.
So, I mean, you have such a wealth of just knowledge and information, just in terms of your research and just what you've done just kind of over your lifetime. I'm sure that, you give advice to, young scientists and aspiring, inventors. I know you've probably even mentored countless students and researchers throughout your career. What advice would you give to young adults who, even children who may be interested in genetics or biotechnology, particularly those who are really hoping to kind of break into these fields, which are just constantly changing and evolving.
Yeah, so I have had over 300 alumni from my students and postdocs pass through my laboratory, and they form a somewhat coherent group. They kind of like each other, and they hang out together at meetings. Um, and I've taught younger kids all the way down to kindergarten, even recently, um, because because they're interested. I think that's the fundamental thing is find something that motivates you, something that gets you up and keeps you roaring all day. Um, and for people like me, I go out and try to find people like that I can mentor. And sometimes you can even reach the ones that are not yet motivated by they can see how much fun their peers are having in their class. Um, but that's the key ingredient right there is being excited about the material, and that's, again, part of the reason that I meet them part way with stories about endangered species and extinct species and, you know, cleaning up the environment, and these are things that they tend to respond to. And also we work with, you know, professional storytellers, novelists and screenwriters, because in the modern world, more and more of that information transfer and inspiration is coming from from essentially from video content.
For those who, may not be, in this space or, even keeping up with it, what would you say is one of the biggest, I guess, kind of myths or misconceptions, from the public that you've heard, within the space of genetics?
Uh, I don't know if it's technically a myth, but I think that I think there's something that's stopping us from adopting genetic counseling and instead investing over and over in very expensive um, gene therapies. Now, I participate in that, but I participate in both. That's the difference. Is I think there's a balance. The gene therapies will be particularly useful for common diseases like aging and pandemics. Um, and I think that the genetic counseling will be much more valuable for severe diseases of youth that are that are genetically inherited. So so there's some kind of disconnect there where people don't see the power of the genetic counseling and instead double down on the very expensive alternatives. When you apply those same gene therapies, which are expensive for rare diseases, they're dirt cheap for common diseases like pandemic and aging, because the denominator, the number of people that are affected, is in the billions rather than in the hundreds, and so you can spread out those costs over a large number of people. While genetic counseling, that there aren't large costs to be spread, so that's a natural. So that's one thing. Uh, I think there are other things that also aren't necessarily misunderstanding, things like anti-GMO and anti-vax. I mean, these are there's certainly an element that it gets amplified, but the GMO foods, you go to the grocery store and it wasn't obvious to the regular citizen what advantage it was to them. And that wasn't
Right.
just a miscommunication. There really wasn't much advantage. And if there's not much advantage, then the risk you're willing to take is close to zero. And the same thing with vax. There is truth to the fact that if you can get by on herd immunity. If everybody else takes the vaccine, then
Mhm, and you
in principle,
don't.
you don't have to. But that's a kind of a selfish way of looking at things. But
Right.
I don't think they're looking at it selfishly. They're just looking at it at the level of understanding that, that they have. So even though it's not necessarily a misunderstanding, I think it certainly benefit for greater education. There are many experiments that have been done over the years where towns or small states have decided to stop vaccinating, uh, and the consequence were lots of kids died, and then they changed their mind. So those experiments are harsh, but they are experiments, and like I said, we're all experimental subjects. We just don't know it. Uh,
Right, we sure are. I mean, we talked a little bit about, more of the technology and how it has impacted. And you did mention AI as well. How do you really think AI is changing the field of genetics?
well, I've watched AI from very early on when it was not terribly effective. Marv Minsky was a terrific visionary in this, and I knew him uh, when I was younger. Um, but recently, some of the things I've seen it do are kind of some of them fall in the category of stunts that are so addictive. It's like we're willing to spend uh, enormous amounts of energy, and this is one of the critiques that's coming up with AI is not only that it's potentially dangerous to us, in a competing sentient beings sense, but it's a danger to us in using up precious energy resources. But anyway, a lot of energy being used to make you know word crafting and image crafting, which basically is hallucinatory. It's not truth oriented. So it's a perfectly valid art form, but there are many other um, applications that require stronger connections to truth. And I think one of those is, back to your question, is genetics and protein design. So protein being the product of genetics. Protein design is probably the best match between AI and any real world thing that I've seen, except maybe art. Um, and it's just working very well. I've recently launched 5 companies that have taken protein design for antibodies, for viral capsids, for editing and so forth, and it really works well, and it works in a particular case that's often not recognized. Back to myth. This is a very high level, almost scientific, is that people emphasize, "Oh, it's computers picking the perfect protein," when in fact, it's computers just picking proteins that aren't dead, or you can generate diversity and then make a big library of those, and you pick the ones that functionally do what you want. And so it's that combination of machine learning plus multiplex libraries they're more powerful together than separately. So I think that's not always appreciated, but that's key to at least all the protein design I've been involved in.
Yeah. We talked about the technology and, just even the term, people use STEM or STEAM, and you mentioned some of that art as well. How impactful have you seen, kind of that A and STEAM and art really play a big part in, either people's, awareness or understanding of how it really helps in the field of genetics?
Right. So So genetics has many components. I mean, there's one of them
Mhm.
which is interpreting the variation from person to person, or the variation even within a family.
Mhm.
Um, and I think that's one application of AI and STEAM in general. Um, Another one is protein design to make protein pharmaceuticals, protein diagnostics, protein materials, you name it. Almost anything that was made in the past without genetics and biology
Mhm.
in the future could easily be made with it, because genetics and biology is the technology that is comfortable through billions of years of evolution, is comfortable with atomic precision. A lot
Mhm.
of things in biology are precise to one atom bond, one
Mm.
uh, fraction of a nanometer. So I think that's where we're going with this.
Yeah.
Fast.
And as we look, to the future of life, how soon do you expect to see significant extension in human lifespan through gene therapy and, maybe other technologies?
Right. Do I think gene therapy is a good one, because it's so it'd be so precise. You can aim it for particular tissues, particular timing, particular proteins. When there's a whole raft of closely related proteins, you can make one of them. You can make new ones. Um, it's very intuitive. So I think that's likely. That you're asking about time frame. In a certain, we've already doubled our life expectancy over a long period of time. But maybe we face a new. We've kind of now running up against. We've exhausted all the things you can do by you know, better hygiene, better diet, better exercise. And now we're getting to the core kind of planned obsolescence as a metaphor for, you know, you, if you're building a car, you might want all the systems to fail on the warranty date, so you're no longer responsible for it as a manufacturer, and you want them all to fail because that means you can cut corners on each of them precisely. And that's sort of what's happened through trial and error evolution. And so we're fighting that. We have examples like bowhead whales that live to 200 years, mice and rats that live 1 year. There's obviously some genetic difference there, and we can exploit that. Plus, our ability to generate libraries to find, and we have found a number of things that impact the aging of animals, and so that's in the clinical trial world. Those turned quickly into preclinical animal trials. Um, and that's what's happening right now is we're taking all the genes that have been found over the years and coupling them together, trying various combinations, and moving them from mice to dogs to humans.
So, I mean, this is such a fascinating area. I know you spent, much of your life, looking at this work. What is something that you are curious about, in this space or even out of this space? It could be something that you really think about, and it doesn't necessarily have to be related to genetics or this space. But what are some things that, may have been on your mind that you've been thinking about?
Oh, yeah, many things. I mean, one of them is how humans are going to merge with or compete with AI. We may need to augment, enhance ourselves a little bit just to keep up. Just to, I think we also need to, pay attention to, um, understanding how our brain works. That will, at a minimum, inspire us to produce AI that's capable of ethical decision making that's compatible with ours, although we barely understand our own, and that we can't get consensus on a lot of ethical questions worldwide. But I think this might force us to be more rigorous about educating alien sentient beings, um, that we show empathy for them, and we show them how to express empathy for us. Um, that's one thing I think about. And then also, when you talk about aliens and talk about space, what is out there? What is it? It seems like life isn't that hard to, we're getting better and better at developing life from non-life in the laboratory. Um, and it starts to make it seem like it's plausible since there's so much water, even in our solar system, there's like hundreds of times more liquid water, not frozen water, but liquid water in our solar system than on Earth, hundreds of times more. So don't you think there's some life in there? And if so, is that life compatible with our life? Is it something that'll just, like mirror life that we just published a paper on, or a whole variety of different lives that will sequester our favorite molecules and turn them into their favorite molecules? And, so we have to be very cautious when we go to these planets, especially the ones that have planets and moons that have liquid water. We have to be careful not to bring them back until we're really sure we know what they are. And we have casually brought back a bunch of stuff from the moon. And I think we need to maybe be a little less casual about that.
How would you say that, your work in this space has, maybe impacted your own personal life? Has it impacted it, or what would you say some of the things are that maybe came up for you?
Well, clearly it affected my personal life because so I spent so much of it on science and education. But there is some self knowledge that comes about um, on purpose or accidentally. I mean, we in order to launch a project called the Personal Genome Project, which was all about testing out technologies in an open system, so you could say which people have which disease and which genetics. So you connect environments, traits, and genes. Um, you can't do that, or you can try to do that on de-identified statistical populations, but it works much better if you treat individuals as individuals, where all these things are clustered the way they would be in a physician's mind. Uh, in that Personal Genome Project, I was asked to be one of the, both the scientist and the research subject, and and so I learned, a little bit more about some of my problems like narcolepsy and found other things were hard to find in my genome, like dyslexia and various other problems. So, um, but we keep searching. Keep getting sophisticated with things like polygenic risk scores. They get better and better every year. Our knowledge of uh, um, neuropsychiatric disorders and maybe even polymorphisms we're getting better at that. Self-knowledge in general is probably a good thing but you just need to have an agreement with yourself and an agreement with your physician that you're not going to freak out on incidental findings. Because there are a lot of things that you can learn that really don't require action, but they're good. It's good to have baseline information so that when you do really get something, you know what your baseline was.
Well, Dr. Church, this has been an amazing interview. I don't know about everyone listening, but I know I have learned just so much in this time with you. And like I said, just really excited because it was something I was really interested in, when I was younger. So thank you for bringing your wealth of knowledge to us. We really appreciate it. And I hope everyone out there has learned something as well. Thank you so much for your time, Dr. Church.
Thank you.
Ignition Sequence is produced by Accelerator Media, a nonprofit organization dedicated to inspiring a lifelong pursuit of knowledge. The podcast is made possible in part by viewers and listeners like you. If you'd like to support our work and help us continue bringing Ignition Sequence and other programs to a global audience, please visit acceleratormedia.org and click the donation button. Your contribution helps fuel our mission to ignite curiosity and empower future world changers. Listen, learn, repeat.